Yeah, hello everyone. Yeah, thank you for coming here. And he's Dr. Pankaj Dhakad. So he did the PhD in University of Edinburgh, and he was working at the lab of Dr. Darren Howard, and he was working on the evolution of immune gene for virus and bacteria. And currently he's keeping the interest in immunology, and he's currently working on the immunology for the mechanical stimulus. So currently he's poking the fly larvae with needles, so he's torturing fly larvae. Yeah, but today he's going to, well, talk about the, well, previous his PhD work. So, Dr. Pankaj, yeah.
Hello, everyone. Hello, everyone, and thank you for having me, and thank you, Yuchi, for the introduction. Today I'm going to talk about my PhD work, which is broadly on comparative genomics and evolution of immune genes in Drosophila. So let me start with the question that you might already know the answer. Why do we study Drosophila? Many of you might be familiar with the Drosophila melanogaster, which is a model organism which have been, like, people are studying for hundreds of years. People in the field of neurobiology, genetics, and immunology extensively study this model organism. But Drosophila genus is just not about Drosophila melanogaster. There are more than 4,000 described species in this genus. And there might be some familiar subgroups and some species that you might recognize, like the melanogaster subgroup here in the green color, and the recently radiated Hawaiian flies, Quinaria group, and there are some outgroups like big Chymomyzas, which diverged from the model organism melanogaster around 70 million years ago. From genomics perspective, there are like more than 500 genomes that have been already sequenced, and they are also publicly available. And out of those, 70 species have the transcriptomics. And this is the key challenge. Only 40 of these genomes have been annotated so far. And for vast majority of the species, we don't have any genome annotations, and we don't know where are the genes in these genomes and what these genes encode. So this creates a huge opportunity. We have this incredible natural experiment. So we have the species that are not just phylogenetically diverse, but they are also like variation, and they are also ecologically very diverse. Sorry. So they have evolved under radically different selective pressures, like thermal environment from tropical to alpine. They have very different life histories, like some species evolve slowly, some are like fast evolving, like their generation time is less. And some species are exposed to more pathogen communities than the others. And some specializes in feeding on rotten fruits, leaf litter, mushrooms, and cactus as well. And so each of these axes represent a different selective pressure. And for the first time, we can now use these genomes to like compare the genomes or the genes, if we can find it, and across this like very large scale in this phylogeny. So for example, we can study the reproductive isolation and spatial genomics, like species that are recently radiated in Hawaiian and Quinaria group. can study thermal adaptation at the molecular level, especially in the species in the Obscura group that are adapted to the cold environments. And we can study the evolution of the dietary specialization. Like you can see, like in this phylogeny, many times the species have shifted from like feeding on the rotten fruit to leaf litter and cactus. And all of these are connected with the immunity, because ecology affects the pathogen community that flies are exposed with. So, and which shapes how the immune systems evolve. So this will be my focus of this talk. So talking about the immunity, so here is the immunity in Drosophila, and like in other insects, Drosophila lacks adaptive immunity, and the immune system is entirely innate. So that means there is no T cells, there is no antibodies. And mainly there are two immune pathways, TOL and IMD. And there are other two pathways as well, but for simplicity, just focus on just these two. So TOL pathways are for the gram-positive bacteria and fungi. IMD pathway is for the gram-negative bacteria. And these pathway contains three classes of genes. First is the receptor genes that recognizes the pathogens in the blood and on the cell surfaces. And the second class is the signaling genes that relays the signals from the TOL, from the receptors and carries it to the transcription factors that transcribes the third class of genes called effectors like AMPs, which directly kills the pathogens. So from here, three questions motivate this studies, like how fast do immune genes evolve? How often are immune genes born or die? And how does the ecology shapes their immunity? So now to answer any of these questions, we first need to find the genes in the genomes to compare between the species. So it is necessary to accurately and consistently identify genes across 300 genomes that have varying levels of genome completeness and assembly quality. Because with poor annotation, we will get wrong gene counts and missing informations if we compare the sequences. So we'll have a biased results. With good annotations, if we have the true representation of the gene counts in the genomes across, and which is consistent across the like all 300 genomes, and we have more complete sequences, so we will have more information to compare, and we can infer real biology out of it. So what is genome annotation? So genome annotation is the process of identifying genes in the genome sequences and like predicting their exact boundaries, like predicting exon intron boundaries, predicting the UTR region, and so on. So a simple way to think about it is it's like finding a bird in a book which is written without any spaces. So, so there is a meaning, but you need an additional information to parse it. So there are three sources of evidence you can use while annotating a genome. Like first is the statistical model, which are trained on the known gene structures, which is like learning the grammar from the previously known genes, and then use it to like annotate another genome. Second is the RNA-seq evidence, so which is like which directly shows you which regions of the genomes are being transcribed. And third is the protein sequences, which tells you like which are the conjugate coding regions in the genomes. So the challenge for this data set is like for 300 genomes, we hardly had any associated RNA-seq data. Only 40 or 70 species has the associated RNA-seq data. And we needed an approach that could work well with limited extrinsic evidence that we have, and while also like take the advantage of the 300 genome that we have. So consider like this is a small glut from the big phylogeny, and we have like annotation for 40 of the genomes. And let's see, in this plate we have the annotation for Drosophila obscura, but for other species we don't have the annotation, and we want to annotate them. And there are some species with the RNA-seq evidences. There are some species without the RNA-seq data. So what we can use is we have the genomes for these species, and we can use the fact that the conserved coding regions is evolve slowly compared to the non-coding region. And we have the genome annotation for one species. What we can do is like project the genome mutations from one species to other species in the clade simultaneously. So now let's look how it works in principle. So we generate a whole genome alignment for a species clade, and from that we get the map of like conserved regions, and we for each genome we get the statistical model predictions, and we know the gene structure in one genome, so we can project the mutations from one genome onto the other genomes as well. And then we have the if we have the RNA-seq evidence for any of the species, or if only one species have the RNA-seq evidence in the clade, we can use that information and we can share the information across the genomes to consistently annotate only the regions that are conserved and transcribed and coding, because we have the protein alignment. So what did we actually get in the end? So let me show you the summary of our annotation effort. So across the 301 genomes, we find that the protein coding number of protein coding genes varies around 13 to 15,000 genes, which is close to the number of genes in the Drosophila melanogaster, which has actually like 14,000 protein coding genes. When we look at the distribution of the CDS length, and we find the mean CDS length is around 1.4 to 1.56 globase pairs, and also Drosophila melanogaster mean length is only about 1.6 globase pair. There are a few things that you can we're talking about here. Like there are some clades which has unusually high gene numbers, like these species, which has around 20,000 genes compared to other species which has around 50,000 genes. And there are some species on the longer branches which has less number of genes. And this whole clade, which has limited number of species with the RNA-seq data at a particularly low number of genes. So this variation can come from the two things. Like first, it could be because some gene families in these genomes are expanding or contracting, or like some gene families are prone to that in these genomes. Or it could be the fragmented assemblies that are causing like the gene models to split, and we are annotating more genes than we saw. But overall, as you can see, the reference species and the Drosophila melanogaster number of protein coding genes and the CDS length sits comparatively with in the broader range of the number of protein genes across both biology and also the serious length distribution. So this means that we are not systematically over or under annotating the genes in these terms. So now we have the annotations. Now we can answer these questions. Just to repeat it, so how fast do immune genes evolve? How often are immune genes born or die? And how does ecology shape immune evolution? So for the first two, I will use the full 300 genomes data, and for the last, I will use the transcriptomic case study. So how does immune gene evolution actually happen, and how do we measure it? First is genes can evolve through sequence changes, like mutations and substitution can accumulate through time in response to pathogen pressure, like arm race with pathogens. And we measure this with the DN by DS. DN is just the ratio of the non-synonymous to synonymous substitutions, and high DN by DS indicates that the protein is evolving rapidly. Second, genes can evolve through turnover, which is like gene duplication, deletion, or genes can arise through horizontal transfer, and there are some genes that can arise through the non-coding regions as well. And these extra genes or the gene loss can give us advantage and can give the advantage for the flies in changing environment conditions. So we measure this using the BirdDat model, and we measure how likely is how frequently gene families expand or contract. So after estimating DN by DS and gene turnover, first we need to account for the inherent properties of gene level genes and the structure of the product of the gene, like which can like independently affect the evolutionary rate. So what I mean by that is, like, for example, a gene has the high which is highly expressed. So in the genome, the genes that are highly expressed are often the genes that evolve slowly, because the mistranslation is costly. And the genes that are longer has the more sites, so there will be more opportunities for substitutions and mutation in these longer genes. And some protein structures that have the sites that have this functional site that are more buried will evolve slowly compared to this the exposed protein structure. And likewise, like if a gene is interacting with more number of has the more number of genetic or protein partners, then it will be under a stronger constraint compared to the genes that is only affecting handful of genes, because these are the genes that acts as a signaling subs in a pathway. So if any changes in these genes could have a drastic effect on the cells. So we account for all of these simultaneously in a mathematical models, and then we can ask, is immune function itself driving the elevated evolutionary rates? So what do we find after controlling for all of this? First is immune genes have a significantly higher DN by DS compared to the non-immune size and position match control genes. So this is in line with the previous studies that also find that immune genes are evolving faster, which is because these are the genes that are in the arm races with the pathogens. But the really interesting story emerges when we break this down by different functional classes of immune genes. And we find that receptor genes and the effector genes have higher DN by DS compared to the signaling genes. And so this makes sense because receptor and the effector genes are the genes that are in the direct contact with the pathogens. And so if the pathogens evolve rapidly, then they need to change and evolve rapidly to counter that. So now the gene turnover, and the result here is quite surprising. We find that immune genes turnover rate is lower compared to the non-immune genes. And at first glance, this might seem contradictory because if immune genes are evolving faster, at the sequence level, why aren't they also being gained or lost frequently? I think this because that these two measures are quite quite different because like the rapid sequence evolution reflects the need of the genes to to evolve with pathogens like in the sequence change sense. But if a gene is frequently gained or lost, then it tells about the the identity of the genes. Like if a particular gene is lost, which is very important in immune pathway, then whole pathway will be compromised, and that will affect not just the protection against one pathogen, but the whole pathway will be compromised, and it will affect many. The effect will be drastic from the immunity point of view. So now, when I break this down by function classes, the picture gets more interesting here, and we find that receptor and signaling genes have a lower rate of gene turnover than non-immune genes, which is expected because overall immune genes have a lot less gene turnover. But effector genes, which has the antimicrobial peptides, have a comparable rate of gene turnover to the non-immune genes. So I think because the effector genes mostly contain the antimicrobial peptides, these are short genes in the genomes and which has only just the one specific role just to kill the pathogen. So the effect on the other machinery in the immunity would be less, and they can afford to frequently gain and loss. If they gain the duplication of AMPs would be much more beneficial for the for a cell because you will have a more opportunity to acquire new function and to kill new targets. So overall, I think this is an important finding. It highlights that many previous studies have focused on short evolutionary timescales and have not accounted for the gene level factors that also independently affects the evolutionary rates. So now I want to move to a more focused case study, like the evolution of immune genes in the distant Drosophila. So there is a fundamental limitation to everything I have described so far. So all of this is was based on the orthologs of the Drosophila melanogaster. And for closely related species, this is fine. But for species that have diverse like 40 to 70 million years ago, then it could be plausible that they have acquired new immune genes and they don't have any homology with other like Drosophila melanogaster and other model organisms in this clade. And these new immune genes would be entirely invisible to our analysis. So we selected 3 distant Drosophilidae, Drosophila difflexa, Drosophila camararia, and Drosophila confusa. So all three species have been diverse from Drosophila melanogaster more than 45 million years ago, and these 3 species inhabit different ecological niches. Like Drosophila difflexa feeds and lives in the east sap flexes, and 2 Drosophila camararia lives in the fungi, and melanogaster, like rodent food and litter. And now to find the new novel immune genes in this species, and to study the transcriptional response in these 3 divergent species, we did an immune challenge experiment. And the first important decision we made was to use the first generation offsprings rather than using the long established lab lines. The reason is that the lab strain can develop an altered immune response after many generations in the lab, while the wild flies should give us the more ecologically relevant like immunity baseline for when we comparing, because we have the distant flies that we want to compare. We want to find the novel genes. So for the immune challenge, we use septic injury with Providencia redclaw, which is a gram-negative bacteria, and it naturally infects some populations of Drosophila. And it is a well-established pathogen to trigger the IMD pathway in these in Drosophila melanogaster. And we infected 5 individuals per species, and we had the 5 matched controls. And then we extracted the RNA and did the sequencing, and this gives us the paired infected and uninfected individual for each species, which can allow us to do the differential expression analysis and analyze how conserved is the transcription response across these distant flies, and to identify if there are any novel immune effectors that are in these species. So what happens when these flies are infected? In both Drosophila species, we see a robust transcriptional response to infection, and we see very canonical IMD pathway immune response. Like you can see, there are like antimicrobial peptides like atacins, sacropins, and diptericins, which are increased around 1,000-fold during the infection. But Drosophila shows an almost absent response to infection. And there are hardly any differentially expressed genes, and there are only 3 known immune genes that are like differentially expressed after the infection. So this raised an obvious question: why did the infection not work, or is the Drosophila difflexa is resistant to Providencia redclaw? So I will come back to in the next. I will come back to this in next slide. But before that, we also noticed that there are several genes that are induced after infection in these species that are unknown, like they didn't have any homology with the known Drosophila melanogaster immune genes. So before that, first let's explore the possibilities of why we see the subdued immune response in Drosophila reflex. So the possibilities could be like these flies are naturally resistant to Providencia redgrae, and they don't need to simply respond to the infection. Maybe they are already infected, so we don't have any real baseline to compare with. Or maybe the infection exterminated bug means bacteria were injected but never replicated, so there was no immune stimulus. So now to explore all these possibilities, we looked at the microbiome data in these flies. So we retained the reads that did not map to the genome of these flies. And then we profiled the bacterial composition of each individuals. So the result was striking. You can see the Providencia reads in Drosophila species, and mostly in the infected individuals, but we didn't see the Providencia in Drosophila reflexa. Instead, we see the spiroplasma in all the species of, in all the individuals of Drosophila reflexa. So let's look at this side by side for clarity. Two things are immediately clear here. First is we see the Providencia reads in the Drosophila species. That means that there is an active infection in these flies and bacteria is replicating. But we don't see the Providencia reads in Drosophila reflexa. Only just 2 individuals had detectable level of Providencia reads. So that rules out this, that the flies were already infected. Because if they were, then we will see the Providencia reads in all infected and uninfected individuals. Second, we find that Drosophila species didn't have any spiroplasma, but Drosophila had high levels of spiroplasma in all the individuals. So spiroplasma is a gram-positive endosymbiont, which is vertically transmitted through the maternal line, and it has been shown to protect the Drosophila melanogaster against the Providencia alcalcipescens, which is a closely related strains of our pathogen Providencia redgrae. So the most likely explanation in this case could be that the spiroplasma is sequestering the iron from the blood, which is independent from the canonical IMD pathway activation to the gram-negative bacteria. So spiroplasma actually is starving the Providencia redgrae bacteria, so they can't replicate in these flies. So they can't. So it is a spiroplasma mediated immunity. But we can't rule out the possibility of field infection. But this is a really nice example of how microbial ecology can alter the immunity of the flies in the in the natural environment, or even can even also replace some canonical immune pathways in the nature. And it is a finding that that came out from our transcriptomic experiment, entirely unexpected, which I find amazing. So now back to those highly upregulated genes that I talked to you about. Could they be novel effectors or antimicrobial peptides? So we selected the genes that were highly upregulated upon infection and that didn't have any homology with the known Drosophila melanogaster immune gene, and has the net positive charge because the typical AMPs has a net positive charge because they targets the negatively charged pathogens, membranes of the pathogens, and the proteins that are amphipathic in nature means they have the distinct hydrophobic and hydrophilic surfaces. And those were the proteins that had the less than 200 amino acids in length. So which is consistent with the size of the AMPs in the most insects. And then we used the machine learning based AMP classifiers like the random forest models, which are trained on the non-AMP databases to predict the probability if each candidate is a AMP or not. And then we also predicted their three-dimensional structure with AlphaFold and looked for the signal peptides using signal phi, which will suggest that if these can be secreted outside the cell or not. So in the end, we had 14 strong candidates and 6 likely candidates. And structurally, these candidates fall into several categories, like the alpha helical structures. So which these are the AMPs that are like sacropins and morocin in Drosophila melanogaster and other insects. And there are some like epidicin-like proline-rich AMPs, which are found in the bees, and these are epidicin that are protect the bees against the gram-negative bacteria. And there are others that are like glycine-rich, which are like diptericins in Drosophila melanogaster, and lysozyme, which are found all across the insects. So the structural diversity here is remarkable. So these are not just the copies of the known AMPs, but they appear to be novel sequences because we didn't find any sequence homology with non-Drosophila melanogaster. So it looks like they have acquired the properties of the AMPs, like a convergent evolution. So this finding of just 20 novel AMPs in just the 3 species strongly suggests that the immune repertoire of the distant flies is considerably richer and more diverse than the melanogaster-centric view. So if we scale this up for hundreds of genomes that we have in this data set, then it will be we will have a much more diversity of AMPs, and we can use that in the therapeutics. So with that, I would like to conclude that we annotated 305 genomes, which is a new resource for the Comparative Genomic Committee and immune. And then we find that immune genes evolve fast in sequence, but are rarely gained or lost, which reflects then an arm race with the pathogens. And we find that distance flies the core immune response, but also harbor novel immune factors, and also there could be novel mechanisms to fight off the pathogens, like I saw, like microbiome, maybe doing the immune work instead. Thank you.
Yeah, I thought that was a super interesting talk. And, I love the idea of kind of breaking from the melanogaster-centric view. And one thing I was curious about is, in the little bit of reading I've done, it shows that melanogaster has a way higher tolerance to ethanol and other, like ethanol compared to other Drosophila species.
Mm-hmm.
And I was wondering if their kind of focus on more of the fermented environment or a more harsh extreme environment could explain them having less immune genes or might bias the type of genes that we see in Drosophila melanogaster compared to other Drosophilids.
Yeah, I mean, yeah, this could be possible because, yeah. And I don't think there are, like, in Drosophila only, I don't think there are immune genes that are like so far, I don't think there are immune genes that are categorized that are not in Drosophila melanogaster. So we don't know what actually the diversity in the other species.
Okay.
And we would expect, because like the environment they are living in is very different. And as you said, like in the fermenting environment, in the litter, like leaf litter, the Drosophila melanogaster lives in is very different from the other species. Like some species lives in the desert as well. Like in the Repleta, they lives in the desert, they feeds on the cactus. So surely they are exposed to different communities of the pathogens, and yeah, I agree. Like they will have different repertoire of immune genes compared to melanogaster. Thank you.
With the novel AMPs, do you have a sense of how old they are and how they're originating?
How old they are?
Like like where in the phylogeny, how deep in the phylogeny they're present, and where did they come from?
So we don't know the origin of these novel AMPs, but yeah. So most of the AMPs were found in two Drosophila instead of the Streptodrosophila difflexa. So which would suggest that, like, if we just take the species phylogeny, then and if they are novel, not just the duplicated sequences which diverse recently, because the structurally they are similar to the non-MPs. I would guess they are not diverse like as far as like the species phylogeny suggests, which is 40 million ago. I think they diverse recently, and because structurally they are very similar to the non-AMPs. But the sequence homology, and also when you compare the sequence homology, these are AMPs which are like very short, and so they might be missed in sequence homology. Like if you have a very hard filters, like 30% similarity would almost entirely like not detect the homology even in the AMPs that are known and in the closely related species as well. And also, like, these AMPs are frequently gained and lost, so this gives them an opportunity to like evolve faster compared to the genes.
I'm just curious, so you did all this work in adults, and do you think that because larvae are immobile and they're rotting food and stuff, that they might actually have worse immune exposures, or maybe like similar profiles? Those vaccines too.
I think larvae would have a more complex immunity in a sense, like the cellular immunity in larva is more active compared to the humoral immunity in adult, and also like they are more, I think they are more exposed to pathogens compared to that, because they are always in the food or like in the nature as well. Like they will always be exposed to more pathogens compared to adults, which can fly as well.
So it seems like very high levels of spiroplasma almost acts like enhancing of the immune response. Is there anything else, any other organisms are known to act very similar? Anybody who else studies immunity flies?
Yeah, there's other organism called Wolbachia, and this Wolbachia mediated immunity protection is in in Drosophila melanogaster natural population, which has been sold previously as well.
Apart from bulbucking,
But, okay. I don't remember it.
So related to that question, so while it's the enhance of the immunity related with the meal killing, so Wolbachia and spiroplasma is kind of a meal killing stuff. So is it related or?
I don't think they are related. Spiroplasma. That's a bad.
Yeah, thank you very much.